Welcome to another broadcast from mindyourhealthcast.com. I decided to take a little turn on some of the heavy stuff we've been doing and give you some material that's a little less heavy, but nonetheless may cause you to think a little bit. As usual, we want to get you to informed as much as possible of what's available, and then it's your choice what you do with it. Now, the broadcast that I have chosen for today is called Know Your SNPs. That's spelled SNIPs like it sounds, but it's not really. It's actually SNP, which stands for single nucleotide polymorphisms. And so, in order to make it easy to recognize and pronounce, the practice has been to put a little I between the N and P and make it spell SNP. And they are so extremely important that it's strange that a lot of people don't know about it yet. As you might know, one of the early people, or groups of people who jumped right into it, were the pharmacological interests and so forth, because they're looking for ways to monetize anything they can, and so it looked like it might be a good market. Now, I'm not sure if any of you have seen one of the back broadcasts, so we did do some broadcasting on DNA and the function of the DNA and some basically information about it. Unfortunately, it seems probably in some cases more complex than it is, and on the other hand, in some cases it's been overly simplified. Seems like there's always those two extremes, and in this case we're going to make it as simple as possible. I want to make it understandable, but yet at the same time simplified. And if you want to get more information, if it strikes your interest, interestingly, once in a while we get something good from the government. And so they published a book, brand new. It's, when I say book, I guess it's a small book or a large booklet. It's about 173 pages on 8.5 by 11. You can go online and print it out, and it's called the Genetics Home Reference, and it has a tremendous amount of information in as much a simplified manner as it can be presented, with a lot of color pictures and illustrations that really are beneficial if you want to know more about what we're talking about. So it talks about genetics in general, and it talks some about SNPs that we're talking about today, and it tells how it works and what it is that it actually does in your body. Now, we've been given some promises along the way, I'm sure you're aware of, that after the whole thing about DNA was first discovered, which was by two gentlemen in England at the time. One was from the US and one was from England, and they were doing research in the concept of DNA. It had been known for a long time that there was information that transferred from one generation to the next, certain traits and so forth. And so bit by bit, the chemistry over time and the investigations finally got down to the point that they realized that in the nucleus of a cell there was some material, and by analysis they came up with a chemical formula and name, which was deoxyribonucleic acid, better known as DNA for short. And when they did that, and they looked at what went on, they knew somehow this was involved in carrying information and coding for things in the body, but they weren't sure what it was or how it worked. And especially they weren't sure of the structure of it, and without knowing what the structure was, then it made a problem to really understand how it could work. And so these two young men in Cambridge, doing research and studies both, were looking at DNA, and at some point it became a common fact that whoever could decipher the structure of DNA would basically give a major inroad and insight into how genetics worked. And so there were people all over the world in the scientific community that were trying to figure out how does these chemicals, because they knew that it had something to do with something called nucleotides, and they had isolated the 4 nucleotides that were present. And so the whole idea came then, how does that organize together into a structure that will tell us more about how it works and how it carries information? And so the race was on, and in the scientific community, when a new idea comes along, there's a lot of scientists who are going for the holy grail of science, which would be to receive a Nobel Prize. And they knew that if whoever could do it would just be assured of receiving a Nobel Prize. So the big contender was coming out of the U.S., and this was a gentleman who had written a lot of books on the quantum combining of factors within cells and within matter in general, but in particular with application to the human body, and that was Linus Pauling. Now, Linus Pauling had actually published a book on the nature of the chemical bond. And he laid down a certain set of rules that seemed to apply, that when these things came together, they would only come together in certain ways. When I say these things, I mean various molecules had a certain way that they would bond. And so he was considered the expert, and many people were sure that Linus Pauling was going to be the one to break the genetic code, so to speak. And so there were other contenders who were hoping that somehow they could come up with the answer before him. As these two gentlemen were researching, they were involved with something called X-ray crystallography, which is a method of shooting X-ray through a crystal of a substance, and then from that crystal they could then determine the structure. By the way the X-rays bounced off of the atom components that made up a molecule, they could then say, okay, this is how they have come together. And so there was a lot of work being done in that area, and they got involved. They weren't primary, but they knew the person who was doing all the heavy research, and so they were looking at all the material that was coming out of that. And so it was the people doing the X-ray crystallography, the two gentlemen who were neophytes in this whole field, so to speak, and Linus Pauling, which were probably the most likely, although Pauling and the crystallography people were probably the most likely to solve the problem. Well, as it ended up, while they were really working on this, someone came running into their lab and said, guess what? Linus Pauling has just published a paper on the actual structure of DNA. And so they thought, oh no, we knew it. We knew he was going to get it. And so they immediately got a copy of his paper that he had published, which was in Science, which is the medical journal in the world, or science journal actually, to publish in, because it's read worldwide and is considered the standard as far as publishing new scientific breakthroughs. So they read the article, and immediately they said, he is wrong. He has actually violated one of his bonding principles in his textbook on the quantum bond. And what he did, he suggested a triple helix structure, meaning 3 winding interlaced backbones connected to the nucleotides. And they took a deep breath, and of course, as it ended up, Linus Pauling became very embarrassed because he had rushed to publish something that he didn't give enough thought to, and it violated his own principle. So that's a little bit of background about this whole thing and how the excitement came. And of course, as you well know by now, Watson and Crick did by cutting out little pieces of paper with the bonding factors on it, and like a jigsaw puzzle on a table, all of a sudden one day, bingo, they hit it. And boy, were they excited, and they thought, gee, we got to publish this right away. And so they published a very short paper in Science demonstrating that this is the actual structure of DNA, which ended up being a double helix instead of a triple helix. And everything worked out fine. Everything joined together as it should. It did not violate any rules, and as a consequence, as I said, they received the Nobel Prize for their work. And so their name went down in the history of science as one of those great leaps ahead. So now what does all of this mean to us? Well, once this happened, there was, of course, a rush to do something with the information. Since they knew that traits could be inherited from one generation to another, they now saw how this information could be coded in the DNA, and from that could then code for making proteins in the body, which are important for the structure and for hormones and for, of course, enzymes. Enzymes are what make things happen in the body. An enzyme goes to an area where 2 substrates or 2 different molecules need to be joined together or broken apart. And so when those 2 get in vicinity with an enzyme, it's almost like it plugs in, gives a big energy charge, and causes the 2 to either lock together and form an enzyme, or a molecule, whatever the molecule is you're trying to synthesize, or to come apart. If it gives a big boost of energy to the point and or drains, I guess the mechanism is not for sure, but at any rate, it causes 2 things to break apart. Now, we're all familiar with enzymes when we think about digestive enzymes. When you put food in your body, it goes through a series of steps, each of those mediated by enzymes that either, in this case, break things down. And for our body, it at least from the food, we break it down. Then when it gets into our body, there are various things that need to be done with the raw material that comes out of the digestive process, like joining together to make things. And so those enzymes are critically important. If you do not have the enzymes, your body won't work. It's possible that by accident things might join together, but the time factor would be so long that in most cases our ability to function much more than maybe lay in a hump on the floor would not be possible. So we need the capability of having these enzymes to speed up the processes. And of course, as I've mentioned before, just something to keep in mind, that all the enzymes are susceptible to 3 factors. One is temperature, one is hydration, water, and the third is pH. All those things have to be correct for maximal operation of certain enzymes. Some have broader ranges of operation and some are much tighter. And so if you don't have that particular, like for example in the stomach, when you eat a piece of meat or anything that's a complex protein, proteins are some of the largest molecules in the body. And the protein molecules are made by chains of amino acids. Okay, so when you eat a piece of meat, you're going to grind it up in your mouth, but it's going to still be in large pieces when it goes into the stomach compared to molecules. And once it gets in the stomach, there's an enzyme called pepsin, which breaks down these large chains of protein into short chains called peptides. And hence the name of the enzyme peptase, or pepsin, I'm sorry. IN used to be the old suffix for enzymes, but they later changed it to ACE. So you'll see some enzymes still referred to, like pepsin, as pepsin, and others called lipase, or protease, or amylase. And so those are keywords that tell you that this is an enzyme. Okay, so that became very important as far as knowing how this DNA could, let's say, supply the information to build protein chains that could either form large complex molecules or, as I say, enzymes, and in some cases hormone factors, would all require amino acids to be, first of all, brought into the cell, then made into chains that when the chains are brought together, they enfold around and create very complex protein molecules. Okay, so those are what make a person unique, is that everybody has their own particular genetic code. And of course, one of the big things they were interested in, how do these figure in terms of illnesses or diseases? If we know the code that could code for something that could create a disease, then we might have a capability of doing something to correct it. And so the idea would be, okay, hey, here's an opening. If we can figure out how to splice a new gene in for a defective gene, because pretty rapidly they learned that genes could be disturbed or mutated. And so mutations mean that it doesn't code properly, and if it doesn't code properly, it doesn't create what it was supposed to make, and hence we have a problem. And so that became, for the pharmaceuticals, of course, wow, if we can figure a way to splice in correct genes, we can, well, they can make a lot of money. That was the big idea. And so that was one of the important things that they thought about. And so before they could do that, they knew that they had to know what the genes did, which ones did what. And so that brought up the concept of mapping of the genes. Okay, so how we want to know what those genes can do. And so they did. They started a mapping process over time. And of course, as the general public listens to the news, they will hear sometimes, oh, well, this we've isolated the fat gene, and we have isolated the gene for schizophrenia, and we've isolated the gene. And so they thought that a gene would code for each kind of problem, meaning if it's a good gene, there would not be a problem, and if the gene was in some way disturbed, then it would become a bad problem. And so mapping became very important. How do we find that? So out of that, of course, came the whole idea of the Genome Project. Now, the Genome Project was, okay, we know there's all this DNA, and one of the early things they discovered was that only a portion of the DNA coded for things, and the rest of it seemed to be inert. In fact, the term junk DNA came up, that this inert DNA that doesn't seem to code for anything, but there's a lot of it there, is just junk. That must be left over from having been somehow gotten spliced into our genetic along the way, but of no value. And as it's ended up, there's more to it than that. I'm not so sure we'll have time to cover it, and it might get too complex, and I don't want to make it too complex, but it became pretty evident that there was a lot of genes which are referred to as introns that did not code in the DNA chains. And so this group that they isolated, they said this is the human genome, And this carries all the information that's needed to help us develop into a human and to carry on the functions of a human. And therefore, we want to figure out exactly what those genes are. And so they started this whole big project, and they had in mind, wow, once we can do this, we're going to have it made, you know. We're going to have a market that's just outlandish, and if you can give people information that will keep them from developing a disease, that's going to be worth a fortune. So it's not only going to be a good subject, it's going to be a big money subject. So just before we get started, because this is going to, as I say, heavily into it, we're going to be talking about some particular aspect of the DNA. So I just want you to get a kind of a picture in your mind. So I've printed out the basic DNA double helix. Now if you look at that, you see those two backbone pieces called deoxyribose. And deoxyribose is made out of phosphorus and other factors, sugar and so forth. And so that's how it gets its name, deoxyribose. And because it is interconnected with nucleotides, it's referred to as deoxyribonucleic acid. Now there is another type that's involved in the whole process of expression referred to as RNA. And that is a ribonucleic acid, and it's ribose instead of deoxyribose. So it's a little bit different, and instead of having a double string backbone, it's just one single strand, and it becomes important in a lot of factors, and many of the things about RNA are beyond what we're going to talk about now, maybe somewhere way down the road, I don't know. But what's important is that you understand, and you can see on this paper off to the side, you have the four nucleotides, adenine, thymine, guanine, and cytosine. These are the four. They show them in different color, and it's those inside the deoxyribose background that generates DNA. Okay, now there's an interesting thing about those nucleotides, that there's four, but they only group together in two pairs, meaning the adenine and thymine, or pretty much always abbreviated AT, adenine and thymine always join together. And the guanine and cytosine always join together. And so that's something to keep in mind, because it's interesting, if depends on which is first in the chain, it makes a difference in the code. Because then the DNA, as it separates to make messenger RNA, which is what carries the information out of the nucleus into the cytoplasm of the cell to make proteins, it makes a difference which of those are on that RNA. So the sequence of the way it's done makes a difference. And so those are the nucleotides that we're going to be talking about a little bit later, so I wanted you to kind of get an idea about it. And as you probably know, the body, every time it builds a new cell, what it does is it copies the information and builds a new cell out of one strand of DNA, or the whole genome. When it makes a new cell, it separates and builds an exact copy for the next cell. And so that potentially is one of those areas where a transcription error could occur, and hence a error could creep in, which then the person receiving those new cells, that person at some point then would start making a mutant variation of the original cell. Because once this new cell duplicated again, it would make it with the error in it. Now sometimes it is done so infrequently, or maybe only a cell or a few cells, that you would never know the difference. But if it would happen that there's a substance that got in your body, for example, a chemical that's mutagenic, that chemical could damage enough DNA that then there could be a problem. And so that's what's called a mutagen, and certain substances such as organic chemicals, even toxic metals could potentially be a problem, but especially pesticides. Pesticides seem to be mutagenic, and some people also say that they're carcinogenic. Now, the difference is that mutagenic can change the code, but carcinogenic can cause a major disruption to the point that it not only changes the code, but it triggers the cell to start making more and more and more replications, which are runaway. And that becomes a cancer. So we don't want to go into heavy detail at this point in those two different ones, but just to give you the idea that what goes on and why it could be a problem. And so there is a lot of information out in the early days, and still highly passed on, that the nucleus of the cell is the brain of the cell. In other words, they're saying, well, that all that information in there is what causes the cell, how it, or I should say, determines how it functions. Now, if you want to find out a lot of information about this, because it's just, as I say, way beyond what we're going to talk about now, but yet understandable, there is a book out by Dr. Bruce Lipton, like Lipton tea, called The Biology of Belief, and he talks about the fact that it's really not the DNA or the nucleus, it's the brain of the cell, that is a library of information. And the rest of the cell, and in particular the membrane, actually controls how the cell functions. In other words, he's doing a contrast with solid state electronics as to how this takes information, the membrane does, and transfers it into the cell to start things happening in the cell. And so therefore the function of the cell is determined by more than just the genetic code. The genetic code is the information that is generated based on the messages or information getting into the cell and triggering a series of responses, which are, again, a bit beyond what we're going to talk about here. You know, there's some new research, and as always happens, people rapidly reach conclusions that down the road things start to come out that show, well, maybe it wasn't like that. As I say at the beginning, it was thought at gene codes for a problem, and that's why if we could splice in a new gene, we could correct that person from having that problem. And especially if we could somehow correct it before they had children, we could stop the problem from being passed on. So that was the early hope of the whole thing. But as it ended up, that didn't quite work that way, and it wasn't quite so simple. And we're not going to get into all the dynamics, but just at this point, let me say that it isn't that simple, because there's a lot of factors involved that are way beyond even the way the body uses the code. And we get into such realms as epigenetics and so forth, which is beyond what we want to talk about today. Because I want to give you enough basic information that you can see the value of the SNPs, which is something that came out of all of this. But I will say that because of this great excitement about the Genome Project, billions of dollars were spent over many years to isolate how many of the genes, how many were there in the genome that was able to do all the things that we needed to have happen. And so then they could begin to map, okay, what makes disturbances and how could we potentially correct those disturbances, et cetera, et cetera. So I'm sure in the news over the years you've heard a lot of newscasts where they would throw in a blip, you know, once we get the Genome Project finished and we know the code of all these issues, then we can start doing genetic therapies, and everybody is going to be healthy and cured, and we will live happily ever after. Now, that is kind of the way it got presented. Unfortunately, as they started the Genome Project and got down toward the end, there were some interesting things occurred. Number one was that they had anticipated that we have approximately 100,000 different proteins in our body. So if you have a gene coding for each of those proteins, that means there has to be at least 100,000 of those genes available. Now, we also know that there are regulatory genes, and those determine how much of something the body might make. That keeps it from running away. In other words, if we needed to code for an enzyme to break down, like a protein or a peptide, into individual amino acids, a message goes to certain cells to start generating some of this enzyme. Okay, so the message goes to the DNA at the appropriate spot, and again, we will not even get into the dynamics of how that can happen, but let's say the message gets to the DNA, so somewhere along the DNA, where the information for that code is, it opens up, and with other enzymes that are present in the nucleus, it begins to synthesize an RNA copy of that particular piece of code that's going to be used by the cell to make that enzyme. And it also is going to have a start and stop factor that will say when to start and when to stop. And otherwise, if it started making a particular enzyme and there was no stop pattern or information, pretty soon the cell would be full of that enzyme, and then the cell would rupture and you would have big trouble. So there is a stop mechanism that's regulating how much of this is made. Now again, how that all comes about gets beyond this, other to say that those are referred to as regulatory genes. And so they figured there's about 25,000 regulatory genes. So now we're talking about a total of 100,000, one for each of the proteins, and another 25,000 plus or minus for the regulator or regulating genes. So when they got to the end of the Genome Project, they were shocked, to say the least. The shock was that there's only plus or minus about 25,000 genes in the human genome. So right away, something isn't right. Where did we go astray? And it ends up that, as I say, it's not that simple. A message from somewhere tells the body to do a certain thing. And so that message gets in, and it can trigger a gene, which can trigger another gene or two, which can trigger another 2 or 3 genes, and those are the ones that actually do it. So it's like the formula has to be assembled from the library of information to make a particular thing. So if that's the case, how do you intervene? Because first of all, you've got to figure out how did that message get there to start this cascade of events happening. And so it was a mind blower. I'm not real fond of that term, but you hear it a lot. And in this case, that's exactly what happened. Now we've got a problem. We're in essence almost back to ground zero again, because we know they're there, we know they code, but now we're not so sure how they code. So as I say, out of that came something called epigenetics and so forth. So the Russians, of course, threw a wrench in the gears to some degree, and I'm not sure who else has done the study, but I've been following a lot of the Russian research, which is in some areas, like, I think, way out ahead of what I'm getting out of the U.S. I spend a lot of time every week on the internet going through a mammoth amount of information and research articles, and I have a room full almost of printed out research studies and so forth that have not even gone in any books yet about the things that are happening. Well, anyhow, the Russians learned a lot more. They learned a lot more, for example, about the 7% of the introns, the junk DNA, that it actually has a grammar and syntax factor to it like language. And now they're guessing that somehow that's why languages around the world have similar grammar and syntax, because it's an expression that comes out of the genetic information that we are programmed with. And there's been cases where the genetic has been changed by modulating a carrier wave that vibrates in the frequency the DNA vibrates at, which is about 58 to 74, 75 gigahertz. And so if you get that as a carrier wave, it's at a station level, like a TV station or radio station, that the DNA can listen to, and then if you can put information on top of that, they seem to have been able to alter the genetic without trying to splice something in. They've altered the informational pattern itself. And I know I've read of a study where they took the DNA code, and we'll talk about the code in just a moment here. I'm watching my time. I don't want to run way off on this. But they were able to take the code of a frog egg that would produce a frog, and they lifted the information off and stored it in a computer hard drive. Now, in a minute, you'll be able to see why that would work very well, because the genetic code does make use of binaries and triplets of binaries and so forth. So they were able to store this information, and then they took a salamander egg, which had not hatched. It was ready. It was fertilized to develop a salamander. So they took a laser beam, and they modulated the laser beam, and they irradiated the salamander egg with the laser beam, modulated by the information from the frog DNA. Lo and behold, when the salamander egg hatched, it hatched as a frog and not as a salamander, so they had totally rewritten the genetic code by what they did. They overrode the original code and rewrote it. Just getting back to what I said earlier about, well, tell me something about whether there's informational or brain-like functions in the DNA. And there's a lot of information out, but again, it goes beyond what we're talking about now, because I'm giving you a lot of this information because we want to get down to the fact that you kind of have an understanding of something that's become available that's tremendously helpful in terms of therapeutics. And that was my whole purpose in bringing this, but it doesn't make sense unless you kind of have some basic understanding. Like I say, if you want know a little bit more, I would look up on the internet this genetics home reference put out by one of the governmental agencies. It's called a handbook, Help Me Understand Genetics. And it was done like 2014 is when it was published, October 9th, 2014. Now that's pretty new stuff. So it's as up to date as they know, and as I say, they mention what we're going to be talking a little bit about SNPs. And so this will kind of fill in behind what I've told you already. Now, as I say, when DNA that is going to build a new cell, it actually builds another duplicate of itself. Okay? And it has these groupings, and I wanted to see that there's groups of 3 that run together. And those groups of 3, I think they probably better show on this next one. The groups of 3 are each a piece of information that will actually code for one amino acid. So that means that when you pull information off of this, each of those triplets in the strand that you copy with the RNA is going to contain groups of 3 of these 4 nucleotides. A and T and C and G, as they're referred to, adenine, thymine, guanine, and cytosine. Those come together to form those triplets. So each group has one piece of information to code for one amino acid. So when it goes into the cell out of the nucleus, it goes into the cytoplasm, the messenger RNA is picked up by something called a ribosome in the cell, and it actually reads it like a piece of informational tape and pulls from the pool of amino acids in the cytoplasm the right ones using another type of RNA called transfer RNA, that the message goes out, okay, we need to make a tyrosine, for example. And so then tyrosine code on the transfer RNA picks up that amino acid and brings it over to the ribosome, and that's inserted into the chain of amino acids that's being built to make an enzyme or whatever it is that's being built. Okay, so those triplets carry that information, and there's charts. I didn't even bring it because we didn't need to know it at this point. But there are charts that actually will tell you, you can look at it and figure out which ones are going to make what amino acids, and if you knew a particular chain, then you would know what groupings of these pieces of triplet code would build that particular protein. Okay, so now we're getting down closer to the SNPs. That's what we're really interested in. Now, the three big uses of SNPs, which is a type of genetic information, is used for something called ancestry. There's actually organizations that if you send in your saliva with some cells in it or some blood, either one, they can run this big panel of it, and they have come up with what they called haplogroups, which are different ancestral backgrounds of different types of people, from England, from France, from Poland, from Czechoslovakia, from Russia, from different areas of the world. And they've already grouped these, and so they will type you to your haplogroup, and they'll even send you a certificate. This is a copy of a certificate from one of the groups that does that, and it has your name on it and the sample number, and then it gives you a haplogroup. Like in this case, the haplogroup is J1, and it says J1 is found at highest frequencies in Middle Eastern and North African populations where it most likely evolved. This marker has been carried by Middle Eastern traders into Europe, Central Asia, India, and Pakistan. The Cohen model lineage is found, which is his name, was a name. The Cohen lineage is found in haplogroup J1. So I don't know what you're going to do with it, but if you want to know something about maybe your lineage going way, way back as far as what group of people it came out of, that could give you that information. Now, it depends on the company. They've gotten the price down. I remember originally when I was looking at pricing, some of the early genetic panels were anywhere from $15,000 to $35,000 to do a panel. So obviously not a lot of people were going to do that. And then over the years it came down to maybe $2,000 or $3,000 to do an extensive panel. And then in recent times, it came down to the SNPs, which we're going to be talking about, which are particular types of things that it'll make more sense when I show it to you in a minute. And the SNPs, it came down to, I think, about the better panel was about $700 plus or minus dollars. Now that's still a little pricey for some people unless it could give you dramatic information, which became more possible. Now I started to tell you earlier that ancestry was one of the uses of it. The second had to do with, of course, the pharmaceutical and drug industry, because something that came up was that if you have certain SNPs that are mutated, it means your liver's capability, because of defective SNPs, cannot detox certain drugs out of your body. And so when you give it to a person that has a particular type of SNP, that person could build the drug up so high in the blood that it could become toxic or even fatal. And that explained why some people would die when they took a drug. The body couldn't get rid of it, couldn't detox it. And all of the dosing that's used in the pharmaceutical industry has to do with how many milligrams per kilogram of body weight that are for an effective dose. And they want to keep it below the toxic dose and or the LD50, which how much of it would kill half the people who took it. And so it's very important to them, but they didn't know that for a long time. One of the last, about, let's see, 5 or 6 years ago, I took a lot of upgraded pharmaceutical training, and for the first time, the pharmaceutical companies in a training program, now I'm not saying they didn't know by then, but as far as a training program, for the first time they're talking about having some of these SNPs, which they have given names of, but actually it's like a code. It's letters and numbers mixed to tell you that it's this one or this one or this one. And so they know then if you have this, then you're not going to be able to take this drug without it at the very least damaging you and potentially could be fatal to you. You just can't get it out of the body. The liver can't handle it, or if it does, it can only do it very weakly. And so if it's weak in getting it out, that person would maybe could not tolerate even more than half the dose. And if it's totally blocked, that means they can't take any of the drug, because pretty fast it's going to build up and be poison. And as I say, their dosing is dependent on keeping a certain level in the blood. Hence, they dose it in such a way that they know the liver is going to take out part of it, and in so many days it would be totally gone out of your body, at least in their estimation. So it's called the half-lives. How long does it take each half to go down to the next half of that? And so it makes a big problem. Okay, so that was the other use, and of course the other is health. And that's the third use, and that's the one that we're interested in, is how can that be useful for our bodies? Well, first of all, we'll show you in bigger picture what a SNP is. If you took just one strand of DNA and you showed at the top what you're starting with, okay, here's adenine as a part of one of the triplets. But when it gets translated to build another strip, instead of it picking up an A, it picks up a G instead. Well, now, you know, if you pick up a G, guess what happens? Only C's can connect with G's. So now you're going to have a CG combination instead of an AT combination. So that alters what type of amino acid is going to be picked to be put into that protein chain. Now, if you're coding for an enzyme and you put a wrong amino acid in, guess what? It doesn't work right. Maybe partially and maybe not at all, depending on the particular one. Okay, so here we show it larger, just so you have an idea. And here is where an error is, and this is what ends up as a SNP. And that's a single nucleotide polymorphism. Okay, so these are called nucleotides. The ATs and CGs are nucleotides. So if there's an error, then an error in one of those nucleotides forces it to join to another one which is wrong, and so that strand of the triplet is now wrong. So it's going to code for a different amino acid, and when the RNA picks this up, that's what it's going to do. It's going to go into the cell and code for the wrong one. Now, sometimes, again, depending on which one it is, it can make a big difference. And so here we're showing how different of these errors can be put into the genetic. There's certain parts of a gene where it can be linked in, where there's no protein production of that, like the introns, and it's not going to have any effect whatsoever. If it gets into one of the regulatory group, and the regulatory have to do with how many copies of this you're going to make or whatever, it's not as much a problem either, because it just means you may not make as much every time you make it, so it only works fairly well. Now, however, if it gets into the third group, which is the coding group, then that means you make a protein that has something wrong with it. It has a wrong amino acid in it to form that protein. So whether it'll carry out its function or partially carry it out depends. Depends on what it is. Okay, so now what we're really looking for in what I'm talking about out of the SNPs is that we're looking to find out if there's a problem with certain SNPs that have now been determined, and there's a bunch of them, have been determined that if that's not working right, it's going to upset the chemistry of your body. Now, I know you can't see this very well, but you see all these circles and lines and so forth. These are different biochemic steps in the body that have to do with what are called the methylation map. Methylation is a process where a particular molecule called a methyl molecule is utilized in certain biochemic processes. For example, if your body locks a methyl molecule on a particular gene, it caps it and it will not express. So we have a lot of genes that are capped off, that are not doing what they should or could. For whatever reason, they've been shut down. Now, they tie that a lot to epigenetics, which again we're not going to get into today, but maybe someday down the road when you're up to par and got all this well in mind, a lot of the stuff that we could talk about would make a lot more sense. Okay, so now why is this so important? Because a lot of these pathways produce things that are necessary, like folic acid goes through a series of steps to produce folates, which are very important in rebuilding cells and the health of the cell. Now, if you have a block of the enzyme that's going to carry out one of these steps to produce the folate that's needed, then you don't produce it, or if it's impaired, you may produce a very low amount of it. If that's the case, you've got some health problems that develop out of that. Sometimes significant, as I say, sometimes nagging, but not fatal. There are some cases where certain things shut down could be fatal. For example, another pathway we have makes ammonia into urea. It's called the urea cycle, and ammonia is one of the things that has to be detoxed out of the body all the time, because ammonia is extremely toxic to the human body. Now, I don't know if you've thought about it or you heard about it, but I'm sure some have heard about the citric acid cycle or the Krebs cycle, which is in the mitochondria of the cells that make energy, but in the liver, there is a cycle called the urea cycle. The urea cycle's purpose is to take ammonia, that is a byproduct of breakdown of amino acids. The body can't tolerate much of it. Now, the kidneys can throw a certain amount out based on certain cycles. Again, we won't get into, because the brain has its own separate little cycle where L-glutamic acid will grab one molecule of ammonia, and that converts L-glutamic acid to L-glutamine, and L-glutamine goes back in the blood, goes to the kidney strips the ammonia off, throws it out in the urine, and converts the L-glutamine back to L-glutamic acid, which goes back to the brain to pick up more ammonia. Because even though the body has a major cycle in the liver that's pulling ammonia so the body in general won't be poisoned, sometimes I shouldn't say sometimes, there are there's a bit of ammonia formed in the brain, and that ammonia in the brain then needs to be detoxed even further, and so it's important that you get that out of your body. Now, so the methylation process involves that ammonia detox cycle. So without taking care of that, you're in big trouble. There's another one called peroxynitrite, which is also very toxic, and that involves methylation process also. And so if you're not making those enzymes to carry out these biochemic steps, you can be in big trouble. And as I say, over time now, they've isolated a huge number of SNPs that can cause all these different problems. And so, why is that important? Well, if somebody has several of these, especially if they get it from both sides of the family, then it's really potent and it shuts it down. So you are never going to have real good health in some of these cases. Let's say you could only partially detox ammonia, you're always going to be partially poisoned by ammonia. That's not good. In fact, if the liver stops the process totally, the ammonia builds up so bad even the brain ammonia detox cycle is not capable of handling that much ammonia. Guess what can happen? You go into a coma. It's called hepatic encephalopathy, which can be fatal. And so these are all important. And energy production, I mean, these are involved in energy production. They're involved in softer factors. Again, as I say, we don't have time to get into detail, but I'm saying all that to say that they have now come out with a test that is so inexpensive that there's no reason why everybody doesn't do it. We are starting doing that in the office. I've been looking at this for some time now. Part of the problem was, so what if you know that? If you can't do anything about it, then there's not a lot of good things you can know about it. I mean, to do with it, other than the fact, well, now I know why I'm sick. Unfortunately, I can't get well. This test provides information of your map, and once we know your map, there's an acquaintance of mine who has a company that makes unique products, and they, along with a very knowledgeable biochemic naturopath, have designed some very unique formulations of herbs and nutrients and so forth, minerals, key minerals, and that all can compensate for the missing information that you need to build these enzymes so that you can get rid of these products in your body and don't poison yourself to death, and that you can function properly, that you don't have a body that's impaired because you can't make good cellular repairs and so forth. And so the test itself has gotten so inexpensive, it's $99 to do a panel. And it only requires, you don't even have to draw blood, you can do a saliva sample with it. And they can get enough cells from the saliva that I'm sure some of you have heard of the polymerase chain reaction, PCR, that if you have a little bit of DNA, there's a way to amplify it so you can build a lot of it so you can carry on a lot of testing with it. And we've mentioned, in fact, Carrie Mullis, who was the scientist, biochemist that developed the process. And in fact, he received a Nobel Prize for that in 1993. And so some of the stuff is pretty new stuff, but to me, so far, this has been the heavy, useful stuff that has come out of all this research, starting way back in the 50s, all the way up till now, that this is some of the most useful stuff at a reasonable price. Instead of paying several hundred or several $1,000s, for $99 you can get a panel. And we have software then that will take what they found and organize it into a nice report that says, here's where you have some big problems. It can affect your behavior, it can affect your energy, it can affect all kinds of things. And it's pretty simple. Those nutrients, sometimes it may have been something temporary because there's some blocks that can occur that are not permanent, but there's blocks that can occur that are permanent, which means that it's always going to be a problem. So if you take this product that will work around that problem, you can get your body up to a high level of health. Now, to me, that's extremely exciting. And when we take this and tie it on with the essential fatty acid information that I've just gotten through giving you recently, when you bring that all together, you have tremendous capability of having a very good high level of health like you probably would not have thought possible. And I'm considering the fact that we're also going to be talking longevity. In other words, you not only will live healthier, but you can live longer healthier. And that's what we are looking for. So, as I say, this is a tremendous use of this information. The fact that, as I say, it was interesting to know, okay, now this is why you got this problem, but unfortunately we can't do anything about it, to know that some very astute people have been able to produce something that can work around this and just make major changes in people's health. I know I've been frustrated occasionally. We run into somebody, we can get them to a certain level, and then we can't seem to get them beyond that. And if they don't even stay on the program I put them on, then they tend to slip back. Well, this is a big part of the reason. This and the real parent essential fatty acids that are real omega-3s and omega-6s in the body, making and redoing the cell walls, because remember I mentioned earlier that information has to come through the cell wall as well as oxygen to make energy and to tell your cell what to do. And if that cell wall is defective, that doesn't happen. That's been established by regular medical research, that cell walls built out of bad fatty acids cannot transport enough oxygen in to really do a good job. And as we mentioned, Dr. Warburg's work way back in the 20s forward, never disputed, I should say never disputed, but never disproven, showed that when you reduce the amount of oxygen going into a cell, you change the function of the cell and it can become a malignant cell. So, with the fatty acids, we can help correct that part of it, and with this information, we can correct the machinery in the cell, all these cycles that are important, and get your body to a peak level of health. So, I hope this is as exciting to you as it was to me, as along the way I've been watching all this happen. I've known about the SNPs for a long time, but there were two things that were a big problem. Number one was, most people didn't want to pay the kind of money to know, okay, I'm sick. Well, I know I'm sick. And number two was there was nothing to do about it. And so now that has been solved, the cost has come down, and the factors that are necessary to get around these disturbances are available. So I hope, as I say, you've gleaned something good out of this broadcast, and we'll be looking forward to hearing from you. I know all my patients are going to, well, I say all, I think most of them are going to want to do this. Many are doing well as it is, but I think that we want to find out what the peak capabilities, so it doesn't hurt to know that, even if we didn't have anything, which I doubt that that's going to be the case. There's always something that can be tweaked a little better and can raise us up to a bit higher level of health. So, I guess our time has expired, and I guess I've given you enough to wrestle with. And I would get the genetics home reference. It's kind of like taking a little brief course in the DNA and genetics and the SNPs and so forth, and it will further fill in some of the things I've told you and reinforce some of the things I've told you, because I know how it is. Sometimes the first time you hear something, you hear bits and pieces, and while you're wrestling with one idea, more information is coming, and sometimes you say, I didn't hear that before. And so this will kind of refresh you. And of course, the nice thing is with this YouTube, you can go back and look again. So that's beneficial. So until our next broadcast, I would encourage you to go look at our website. We do have the page up on fatty acids now, the essential fatty acids, and I will be putting something in there on this information related to the SNPs. Otherwise, until our next broadcast, be healthy.